Thank you so much for the opportunity to share a little bit with you about Mission Bio. Mission Bio, as a company, we're actually a relative newcomer to the community of regenerative medicine or cell and gene therapy. But we're super excited about our platform, which is quite unique. We can offer high-throughput single-cell DNA sequencing as well as DNA-based multi-omic analysis. And we truly believe that with the power of our platform, we can really enable the drug developers, in particular in cell and gene therapy space, and really to fill the gap or void that exists today with much needed deeper analytical characterization of your drug products. We can actually fill that gap. And with that, we can help to accelerate your drug development process, shorten that, as well as to ensure higher probability of success for clinical trials and the long-term patient success. So we are based in San Francisco, the city that is almost just as windy and cold as today in Barcelona. We are relatively young. We spun out of a microfluidics lab out of UCSF. And really, our expertise is actually quite complementary to many of the cell and gene therapy drug developing companies. What we have is a group of scientists that are specialized in microfluidics, in engineering, in molecular biology, which translates to specialized assay development, as well as very talented bioinformaticians. So what we can offer is really translate the power of microfluidics platform to answer very specific questions in a total solution. We have deployed the platform since 2018, so we've been commercial for about 4 years. Our first foray into the commercial world was actually focused on oncology, to really utilize that higher sensitivity of single-cell resolution to better understand the coexistence, co-occurrence of mutations that today, through bulk sequencing, we actually miss that information. We believe that's super critical for understanding drug resistance to really help taking precision medicine to the next level. So where are we as an industry for precision medicine? If we look at the two large disease areas that really have benefited from the last 20 years of technology evolution, on cancer, there's dozens of targeted therapies that have become commercial in the last 20 years because of our understanding of the human genome, our understanding of disease. But I really wouldn't say that we're hugely successful yet, is because more than half of the clinical trials still fail. And the failed is not just clinical trials, but actually does impact patient life and our lives lost. That translates to a very significant number of our patients still succumb to cancer because of drug resistance. And more than 85% of those are because of the uncharacterized co-occurrence of mutations that either are adapted to the disease treatment or actually were just went undetected. And the second part, I love the word of rare disease, right? We all talk about rare disease, but the combination of 7,000 of those accounted for hundreds of millions of people that actually get inflicted by this rare disease every year. More than half of those are children, and a good percentage of those children don't live past the age of 5, and 80% of those are genetic, and vast majority of those don't have actually approved therapeutics. And this is why we're here. Why we're here with this community with cell and gene therapy. We believe we have on our hands transformative technologies that can actually address both of these largest categories of deadliest diseases, cancer and rare disease. If we look at all of these things, right, the cancer, genetic disease, and cell and gene therapy, they do share a couple of things in common. One is that they're all very heterogeneous. They're not just a monolith, using one of our SAB's members' words. And the second one is that it's all genetic in nature. There's actually DNA mutations, DNA changes, either SNV or copy number, or it changes we are actually trying to introduce to these heterogeneous cells. And so understanding those changes at the DNA level, as well as the phenotype associated with the genotype at the single cell level, is going to be extremely powerful tool. So in the center is really what we do offer, the Tapestry platform, where we can better help you to characterize the disease, characterize the therapeutics, which can translate to, again, more successful therapeutic outcome. And the interesting part between the left and right-hand side of the diagram is that they're also related, because many of the rare disease treatments or cancer treatments today with cell and gene therapy the adverse events actually more or less are correlated with potential transformation of the engineered cells in the patient body. So it's really about understanding cancer, and are those cancers also related to what we have introduced. So I want to talk a little bit about being a little bit geeky over here on the mechanism behind why Tapestry can offer that single-cell DNA analysis. It's not by coincidence. Actually, the team from UCSF went to design this microfluidic system with the purpose of getting to the DNA changes. And so it's actually very, very creative, very innovative design. What we call it is a two-step microfluidics. On the left-hand side is really cells or nuclei or other things in suspension can be pushed into the channels of microfluidic channel. Each channel, the diameter is a little bit thinner than a diameter of your hair. So in this first step on the left-hand side, we generate these droplets that encapsulate each single cell. So now you're creating tens or hundreds of thousands of independent reaction vessels, each of which contains a single cell. In this first step, we also introduced what's called protease. And so the protease function is to actually degrade away the chromatin structure that wraps around the DNA. So that's actually a very key step to actually unravel your DNA. Now your DNA is exposed. You can actually access any parts of the DNA for sequencing or copy number analysis later on with PCR. We then inactivate the protease, and then the cells get pushed, the lysate gets pushed into merging with the second droplet, which contains a barcode, which now each of those cells gets a unique oligo tag that is integrated into the amplified DNA fragments. Then the final outcome is actually readout is on NGS platform, for example, Illumina NGS platform. We can also integrate protein analysis by adding antibody conjugated with oligos. So then that translates proteomic readout to also consolidate on NGS. So we only have to read sequences associated with specific barcodes. It's quite nice. So in this way, we can analyze up to about 1,000 different amplicons, as well as up to about 100 different proteins. So it's really at the center of the genomics and proteomics at the single-cell resolution level. So we ultimately do believe that we represent the ultimate platform that can take precision medicine to the next level. As I mentioned earlier, cancer was really the first area that we have gone into, and is really manifested by the increasing number of publications that have come out. Quite excited. We feel we're at the inflection point this year with a tremendous new number of publications in New England Journal of Medicine or Nature that really highlight using the single-cell resolution can help us to better understand drug resistance and better actually have innovative therapies to cure cancer. But what's the relationship with cell and gene therapy? Cell and gene therapy space, without me talking about it, is we are here is because of the great promise, not just by ourselves, but also by the regulatory bodies and our expectation and anticipation of the future success of cell and gene therapy, ultimately to transform how patients are treated. Not just FDA, we also heard at the conference that the European Medicines Agency also anticipates about the same number of ATMPSs being approved each year starting 2025. So that's the same number FDA has projected. What it really means that is a lot of work for all of us. Is only 3 years from now, we're going to increase the approved therapeutics from today's a handful to actually 10 to 20 of those on each continent. And that's tremendous work. How do we get there? There's actually 2 specific areas we think are super critical for us to solve for as a community, for us to support you as your ecosystem. And the first is to accelerate your development cycle. The shorter development cycle is, the faster you can actually get into clinical trial. And the second is to actually have the right tools so you can maximize your success of your clinical trials, therefore the ultimate commercial outcome. Both of which are actually deeply, deeply related to our ability to help you to better characterize your therapeutic products. And that does come back to single-cell analysis. This cannot be accomplished alone by yourselves, by ourselves. It's really in combination as a community. As we've heard in the past couple of days, there's tremendous, tremendous effort from the agencies, from regulatory bodies, looking at how to better provide guidelines so we can move faster as an industry. We're very excited to see FDA taking bigger steps in this manner with the more recently released in March, the guidelines about looking at really focusing on the design platforms for these edits to look at the components by focusing on on-off target, as well as looking at potential longer-term monitoring of potential adverse events of these patients, i.e. cancer transformation. And so this is where we feel quite strongly that we can be your strong partner to help you answer any, most, or all of those questions so that you can get faster development and better outcome clinically. We always get asked the question, so what do our partners do today? Our partners do today is really a combination of many classical platforms because you have to characterize your product in so many different ways. You have to use many, many different ways to try to push the data together. And I would say it's really not just platform choices. I think what's really here, an inefficient part of this overall process is the opportunity cost. Because each of our platform companies, the drug developers, they're doing their homebrew assays. You're actually spending tens, millions, or millions of dollars to try to develop an assay on your own. And so what we're offering here is that we're hoping to leverage Tapestry platform and begin to offer that end-to-end solution that is already designed for your purpose. And so that in partnership, we can help you to reduce the cost that you have to invest into analytical platforms, and in combination, we can help you to get there faster. So these are just some examples on what kind of questions Tapestry can help you answer. Really, in general, between the two different categories of gene transfer and gene editing, we can offer tremendous value. On the gene transfer side, you can look at the diagram here. We can actually very accurately quantify transduction or transfection efficiency. We can look at a vector copy number. I'm sure these are all very familiar questions that you think about all the time. And as well as looking at the genotype and the phenotype correlation. And very similar set of questions on the gene editing. On top of that, we can actually quantify the chromosome loss or translocation, which can have a long-term impact on the patient health and the patient overall disease outcome. So these are just really the beginning part. There are other questions we actually believe Tapestry platform has the potential to solve for, like integration sites. And those are really things that we continue to innovate, and we hope to deliver to you a full solution that can help you to accelerate. What's more important is actually all of these questions and answers can be combined in a single workflow. We can actually combine the panel design in one assay, so you don't have to have disparate assays on disparate platforms and try to deal with multitudes of different sets of data. So what are the alternatives, right? Alternatives, there's really two paths without Tapestry. On the first, I'll go through each of them separately. The first is bulk. If you don't have the single cell analysis, you have to analyze your drug products in bulk by, for example, NGS directly. But obvious, what you're losing the information is the population distribution of your heterogeneous drug products to fully understand the safety as well as characterization of that. You do have other options. The other option is potentially to outgrowth your cells in, say, 96-well formats, and then with the clonal expansion, you then go analyze them on different platforms. That usually takes weeks, months, if the cells do grow, and you still have to spend energy and time to design those assays, and actually that in itself gives you tremendous level of high variability. So what we do offer here, if you do the single cell analysis, you can bypass the cell enrichment, bypass the clonal expansion, and actually get the results in days other than weeks. To give you some more specific use cases of a Tapestry answering some of these questions, for example, vector copy number. On the left-hand side is the bulk data, which gives you an average of 3.5, which is not very meaningful. On the right-hand side is a single cell data that gives you the distribution of the number of copies and that component within your heterogeneous cell population. That gives you much more comfort in understanding, do you have a cell population that have copy numbers that exceed 5, which is deemed to be unsafe. Genotype and phenotype is also another tremendous application that can have huge impact in monitoring whether your CARs are expressed, or whether your cells are differentiating to the cell types that you desire, whether these cell types actually persist in the patient body when you can analyze patient-derived materials directly, looking at the genotype edited cells and what's the outcome of the edited cells. And all of those are going to be delivered in actually reports that are meaningful to you in the manner that can be interpreted, can be used directly, incorporated to help you shorten your development cycle. So this is where we actually do add value, is actually the entire value chain, from your development, preclinical, all the way through clinical trials, and hopefully we can also help you to ensure your patients are safe over the long term. So how do we deliver that to you, right? There's actually many different formats that the solution can be delivered to help you. The first is actually, in addition to products, we do have pharma assay development services. This is for our partners who actually don't have all of the bioinformatic expertise in-house. You don't necessarily want to go and build out that entire team to run the platform. You can actually access the assay, give us the samples, we'll provide the reports back to you, and actually could work with you side by side, scientist to scientist, and tailor the assay to meet your needs. You can also choose, obviously, to buy the platform, so you can homebrew, and also can enjoy the fast turnaround time, as we talked about. You have the platform in-house, you can actually get the assay results in a week, as opposed to waiting for others to deliver results to you, which may take weeks. And we also can do technology transfer to the CDO or CRO or CDMO of your choice, so they can actually carry out the assay to support your autologous or other patient monitoring as part of your clinical trial, so you don't have to go to different partners. At the end of the day, when you do have great commercial successes, you are going to want the more distributed clinical centers, medical centers, to be able to help you to monitor the patient progress. And we do have over 200 install base today already in various comprehensive cancer centers that we believe can be the long-term solution for you, so that your patients can get that ongoing care. So we already have a great example. Our very first CDMO transfer was announced in February with Avance. And so what we did was we transferred the transduction efficiency assay to them. So now they have actually implemented that in their CGMP facility. So they're actually carrying out this as a part of clinical trial for their customers. So we're very excited about being a part of their success, therefore their customer success. So this is, in a nutshell, of what Mission Bio offers. Is really, we believe we can help to shape the future of precision medicine with our development, accelerating your development timeline with our single-cell resolution, and with being a very strong partner to your future success. And coincidentally, when I landed about 2 days ago, when I logged down the Wi-Fi, right? So the Wi-Fi password for the conference is Forge for Hope. I really think it's coincidence, because Forge is really create. It's really about shaping the future. And so here, we're just proposing that we're here with you. We want to co-create that future with you. We want to co-forge that incredible hope to offer to the patients, that we can help to make something impossible possible. So thank you so much for your time. We do have our booth outside, and our colleagues are here. Hopefully have meaningful conversations with you to forge that future partnership. Thank you.